Okay, so thank you so much for the introduction, and Alex and Alice for the invitation. It's great to be here. I hope everything is working well also in remote. And yes, so I'm very happy here to present the work that we have been doing in my lab to try to mine what we call the hidden diversity of the human microbiome. So in this presentation, I would like to first of all introduce you to what is this hidden diversity of the human microbiome, then try to find out how we can analyze it and uncover it, and then how we can use it for real problems that we want to solve. So if you are here, I don't think I need to introduce you to the human microbiome. But again, the human microbiome is far from being understood. There are many different subtle points and aspects that really need to be understood to have an impact in biomedicine. And although there are already a lot of potential both in the field with the available tools and ways to mine the microbiome, I think we need to continue focusing. We need to really understand and embrace the complexity of the microbiome before having a real impact in the field. And so today, there are several ways in which you can study your microbiome. We focused in my lab for 90% of it, I will say, on metagenomics, short-term metagenomics, which is briefly summarized here. We start, of course, with the collection of the microbiome sample, and already this can be a quite of a tricky operations in some settings, but we are not going to go into these details here. Then there is the step of isolating and extracting DNA. So ideally, you want to free the DNA from all the available cells, but there are many different kinds of microbes inside there. So this is also a very difficult and somehow biased operation. Then you need to do the sequencing, and this needs to be really high throughput sequencing because you need to access many genomes at the same time, not only those that are very highly abundant, but also those that are less abundant and so more rare in the community. And you end up with big data, making a long story short, with millions and millions of short reads for each sample. So we start from uncultivated samples, we get to a metagenomic data set of short reads, multiply billions of times most of the times. Here is where the computational pipeline starts. First of all, is the somehow usual preprocessing pipeline to try to clean your data, but also to remove contaminants. And in this field, the contaminants is actually our genome, the human genome, that not only the human genome, but the human genome is definitely something you need to get rid of. This can be quite simple, just mapping against the human genome and remove whatever maps against that. What gets interesting is this second step that we call it sequence analysis or primary analysis, and I will focus most of the talk today on it. Let me see if I have a pointer. Maybe I need to, sorry, taking a pointer here. Yeah. So this is a set of operations for getting from unstructured sequence reads to something more understandable. And these more understandable features can be which microbial species are present in each sample at which relative abundance. So quantifying the microbes that are present, not only bacteria, also archaea, viruses, and fungi. But you may also focus on which functions are present, which enzymes, which pathways, which functional modules, microbial functional modules, of course, are present, and with an idea also at the relative abundance again. But you can also try to reconstruct the full genomes, or at least pieces of the genomes of the microbes or present in the community to then do some sequence level analysis. And these set of features are then provided, and you can link them with the clinical data you may have on your data, who is disease, who is healthy, whatever. And this is more classical, if you like, bioinformatics analysis, at least in theory, trying to do something like biomarker discovery, meaning finding which species, functions, or genomes are differentiating to different conditions, for example. But there is much, much more, and we will touch base on a couple of these points later. Extremely important for me is that the human microbiome is extremely variable from person to person, from population to population. So is very, very important, again, to do a validation analysis. Whatever you find in a population, you should try to replicate it in another population, because there may be some confounding factors that are not able to be modeled in the first population, and these will lead to a lack of reproducibility. Is not only validating in other populations, is also validating similar conditions, because again, many conditions may be confounded, but with many other conditions. All right, so having this in mind, let's deep into some examples. So here is the results of sequencing and assembling a skin sample. So we took a skin swab, we extracted DNA, we sequenced it in high throughput, and this is one single sample after doing assembly. Assembly, probably you know it, means trying to somehow overlap the reads to reconstruct longer sequences. And each bubble here into this plot represents a contig, a reconstructed contig from the community of mixed, of course, microbial species. We took the skin in this example because the skin is slightly less complex microbiome than the gut microbiome, for example, so it can be somehow visualized. So each bubble is a different contig. The size of the bubble is proportional to the size, to the length of the contig. And then here is representing that the bubbles are put on GC content against coverage. GC content, each single bacterial species has a different typical GC content, and coverage, something that is more abundant into the sample, will have an higher coverage. And so we plotted this. And then what we did was mapping the contigs against the full NCBI and coloring it based on the bacteria that it eats. So up here, we have Cutibacterium acnes, a bacterium that we all have in our skin, very abundantly, is like almost 80 or so X coverage. This is the typical GC content of Cutibacterium acnes. Here we have Staphylococcus epidermidis, but what I wanted to focus on this slide is all the white points, white bubbles here, that are representing contigs that are not matching against any known bacterium in NCBI. However, you can do some clustering analysis, clustering and binning in this case is called. So for example, you can discover that all these bubbles here have some signatures through which you can cluster them in the same bacterial genome. And if you are able to do that, then when you have a full genome, even though it's a genome of an uncharacterized bacterium, you can do phylogenetic analysis or other kinds of analysis to try to understand what is the context, at least, of this bacterium. And in this case, we can place this into the phylogeny of bacteria, and these, together with other two samples that we found in the same study, is placed here in between two families of bacteria. Each single point here is a different leaf node, here is a different species, and in color there are different families. So here we probably discovered, in this case, a new species of bacteria belonging probably to a new family, or at least a new genus of bacteria. So this is one of the unknowns in the human microbiome, species that have not been cultivated so far. Microbiologists were not able or didn't focus on isolating and characterizing them, so they don't even have a name. But there is more hidden diversity. So this is a map, I'm telling you in a second what it means, of Escherichia coli, the most well-studied bacterium. And each point here represents a different strain, a different genome of this bacterium, collected from gut microbiomes around. So basically with the same thing I showed you before on the skin, but on the gut, and this is specific of Escherichia coli. The point here is that you don't see any 2 points here that are exactly the same strain. So each of us, probably 60-70% of us, has Escherichia coli in the gut, but is usually a very different strain. And in fact, coming from another viewpoint, if you like, the pangenome analysis, so looking at which genes can be present in strains from a given species, we know that E. coli has an open pangenome, and most of bacteria have open pangenomes, meaning that every time you sample a different Escherichia coli, that Escherichia coli will have from 5 to 10 or even more genes that were not present in any previously sampled Escherichia coli. So, an Escherichia coli has a genome of around 5,000 genes, but the pangenome is much bigger, is 5 to 10 times bigger. This means that, even if you sample a lot of strains, like in this case of Escherichia coli, again, the most well-studied bacterium, you still miss many genomes that can be present in the species that are not in databases. So overall, this means that if you take a human metagenome, so a sequencing of a gut sample, for example, from stool, and you map it against the full NCBL database, which is in any way a bad idea because that will take ages. But if you do that, and we did it for some samples, you map probably 60-70% of your reads. All the rest is what we call the microbial marker, which is coming again from unknown species, part of the pangenomes of known species or unknown species that have not been sampled yet, and also, but this is not the point of this talk, from viruses and fungi in some cases. Of course, there is a problem also of representation of populations. Our population, westernized population, are quite well sampled, so there are many more reference genomes for our population. If you look on non-westernized population, the mapping is much lower than that. So this is a problem of the unknowns, unknown species, unknown genes, and these may lead us to miss a lot of key features in studying the human microbiome. So what you need in this case is to clearly move forward from what the 16S RNA gene sequencing can do for you. Maybe some of you are familiar with that. This is a technique in which you are amplifying only one gene present in all bacteria, and so you can have an idea about the taxonomic characterization of your sample, but lacking resolution, taxonomic resolution, and completely the functional potential. You can go to the species level, understanding which species are present, but this is limited, again, because you miss intra-species diversity and the species that you don't know. So what is really important to do is what we call strain level profiling, which we and many others are doing. This is our publication in the field, meaning that it's a long process. We started 10 years ago to try to answer and to these main 4 questions. First of all, identify whether in the sample we have a strain of that we already know. And this, I can tell you, is not a very important task because it's very rare to find in a microbiome, in a new gut sample, a strain that was already present in a database. So much more important is to discover new species and new strains directly from metagenomics, also characterize them functionally, so I'm having an idea about what could be their role, and also I will show you later how to track transmission of these species. Very importantly, also, I told you about the variability of the microbiome. Let me show you an example here, try to reinforce this. So this was a study that we did in trying to understand from stool metagenomes whether someone has colorectal cancer or not, with samples taken even before colonoscopy, so before the diagnosis. This was a study done in Milan. We were quite happy. The species are there. And if you classify them with respect to disease, non-disease up here, you find some biomarkers. Great, these are the biomarkers that we found. And then we said, okay, let's go into another city and do the same. Turin is not that far from Milan, and these were the results. And however, the results, the biomarkers were quite different. So is this true? Is this not true? Well, this is the problem of the variability, because when we went back and included many other studies, some from us, some from literature, we did find several biomarkers up here that are biomarker only of one data set. These are things that are probably not directly related with colorectal cancer, or they are related with colorectal cancer in the subtype, in the sub-conditions of each specific code. However, if you look down here, there are some species that are reproducible. These are the biomarkers that are reproducible across these 7 or 8 different cohorts. With all of these are a set of 7 to 10 typically oral species enriched in the gut microbiome of patients with CRC in all the studies that we looked at. And so it's really important to look at analysis. And here we have codes from at least 3 different continents. And I will show you later, we further increase these set of samples. So it is important, and this is the second methodological key, as I see it. The first was strain level profiling with those four goals. The second is to be able to profile the microbiome at the global scale. And we cannot think of doing, a worldwide sampling for each study we have in mind. We need to integrate existing data. Luckily, or is a feature of the metagenomics, it is quite possible to reuse all data when available metadata is available, because the sequencing technology is pretty the same, and there are ways to integrate data, especially in the meta-analysis way, in which you don't really need to integrate the raw data, but you integrate with late integration, let's say. And this is just an example, but these are these are a bit outdated, but more than 100 publications with quite extensive metagenomic data, each of these publications that we collected and profiled with the framework I showed you before, in the same way for all of them. And we produce an R package in Bioconductor, which allows you to just download all the profiles of these samples. So if you have a new study, you can sequence your samples, profile your samples with the same techniques, and then put your profiles, your sample, into the context of the worldwide microbiome profiles with, of course, a lot of information about, for example, disease, non-disease, age, sex, and many other features. So you may want to slice, this amount of data for what is of interest for you. For example, matching your data set for age, sex, and BMI at least. And this is something that we are pushing forward. But so this was a bit of an introduction about the need of these methodologies to dig more into the bottom of these microbiomes. So let me show you a little bit more than just that skin sample on how to profile unknown species in the human microbiome. And just as a reference, we are looking at this part here, in which we are doing assembly-based profiling of metagenomes. And indeed, what we did was to basically repeat that operation that I show you on a skin sample, but on almost 10,000 metagenomes, including skin samples, but mostly stool metagenomics, because the field is a bit skewed toward the gut microbiome. And when we did that, technically it's called single sample assembly, we retrieve more than 150,000 bacterial genomes that we could define at least medium quality. And these 150,000 genomes, of course, many of them were coming from the same species, but the taxonomic characteristics of many of these bacteria are quite badly characterized. So what we did was to cluster de novo at 5% average nucleotide identity distance all these 150,000 genomes. And these ended up in a catalog of almost 5,000, what we call species-level genome beans, representing the species-level diversity of our microbiomes. And this is a bit of the details of the approach. I'm not going to go into the details. The most important thing is that there are very good assembly algorithms available. However, the key point is to do quality control of the genomes, because, of course, metagenomic assembly is a very difficult operation and is easy to fall into chimeras and other problems. And yes, so we had high quality genomes, medium quality genomes, and this is the key part of clustering genomes into species level genome beans. You may say 5%, how you choose the 5%? Well, 5% ANI is the typical distance of taxonomic characterized bacterial species. Is surprising how it is a good fit for these phenotypic based taxonomy. Of course, there are issues in that, but it works pretty well, and I can show you some data later. Anyway, this is the phylogeny of these 5,000 species. So this is what we can call the microbial, the human-associated microbial tree of life, with the 5,000 species, one genome each species. And what is the most important thing here is that almost 3/4 of these species-level genome beans do not include any available bacterial genomes in NCBI. So we cannot name 75-77% of these species, because they represent sometimes also pretty prevalent species that have not been cultivated and named so far. It is true some of these species are quite rare, showing up maybe 3, 4, 5 times in different individuals, so maybe not that important, but some of them are very prevalent. For example, if we zoom in into this part of the tree here, we are in between two quite well-studied bacterial genera in humans, Ruminococcus and Faecalibacterium. And inside here, you can see there are many different species-level genome beans, including thousands of genomes that have not been characterized. Inside here, there are only 2 reference genomes, A and B, that are not matching most of these species. For example, the red species here includes 1.8 thousand. Now we have more than 4,000 genomes of this species. This happens to be the 7th most prevalent and abundant species in our gut, and the closest genome is this guy here at more than 20% average nucleotide identity. So it's really a different species. It's probably actually a different genus as well. So this species was quite well studied by us. We call it as the name of our institute, actually. And you can even look into the phylogeny, the strain-level phylogeny of these new species. Without going too much into details, but around here you can see the color based on the country in which the original sample was coming from. And we have a subspecies in non-westernized population. It seems there is also a subspecies quite specific of the Chinese population. But you have the genome, so you can also look at the functions, and this is what you can do with simple gene presence absence analysis, and you can indeed find that some of these strains, some of these subspecies here, have different functional profiles and functional and operons actually showing up or not. So, with this work, what we did was that we were able to increase the mappability of the microbiome to probably 90%, more or less. And this is very important because you can make sense of the unknown species. Importantly, this because given that we sample a lot of non-westernized population, we increase a lot of non-westernized population, which is important to have a diversity of microbiomes. However, with assembly, you cannot see species that are low abundance. This is because, for assembling a genome, you need at least 1x coverage at the very minimum, but there are other problems in assembling genomes. So it's important actually to go back to compare, to get reads, map against genomes and make sense of them. If we are able to do that, what is needed is to be able to reconstruct a genome only once in a very large population, and then be able to map reads against that genome, even in metagenomes in which there are few reads coming from that species. So that is what we did. Actually, the 150,000 genomes become 1 million genomes recently. The work I showed you was a few years ago. Now, again, we expanded to almost 1 million genomes. And in this way, we obtain many more species-level genome beans. I think we are around 70,000 species-level genome beans now, not only for humans. We also included many environments, from water environment to soil and so on and so forth. And the problem then was that mapping against 1 million genome is really unfeasible. So the trick that we use, and this is done in our software called MetaFran4, is to look for genes that are specifically characterizing each species-level genome beans. So genes like this one here, that is present, for example, this guy here is present only in SGB number 1 and is never present in the other SGBs. If we do that, we can have specific and unique markers that are a unique barcode for that species. So if we find reads mapping against these kind of markers, well, we can say for sure that that corresponding SGB is present. Of course, we don't do that with one genome per species. We do that with hundreds of genes per species to have, of course, statistical power in the identification, but also in the quantification. So in this way, you can end up with a very quick algorithm, not because the mapping is particularly fast, but because we reduce a lot the data set to only only 200 or so markers for each species level genome bean. So it's like 70,000 multiplied by 200. That is the maximum number of genes you have to map your reads against, again, instead of doing 1 million genome mapping. This is quite successful. We validated it in systems like Kami, and it works very well. And there are some environments, including a human tract, in which you find more than 40, on average, unknown species in a given sample. But other, environment like ruminant, for example, they have many more unknown species than known species, and it's extremely important to be able to profile them, to the point that the mappability was increased for example, non-human primates to very, very few species to more than 25% of the species that can now be well characterized. This is a big framework that we are working on. One of the operations is taxonomy profiling, but in the map before I show you that there are many other operations. And for those of you available with our framework, I describe you Metafran 4, which is the evolution of this software here, but we have many other tools for strain level profiling and functional profiling, which I don't have time to discuss today, but we are updating them also based on this idea of the species level genome beans and the power of uncovering the unknowns. Let me show you these tools a bit in action. So in this heat map here, instead of having samples, in each column is a different study, and the color is representing the prevalence of a given bacterium in these datasets. There are many datasets for a total of more than 10,000 metagenomes here, and the main difference here is that above here you have westernized population, so our population, non-westernized population with a very different lifestyle, diet, access to drugs, and so on and so forth. And we also have samples from ancient stool samples. These are called coprolites, and you can retrieve the microbiome out of them. Above here, you see the species that are very highly prevalent in non-westernized population and in Asian populations, but they are not almost anymore present in our gut microbiome. You know, light blue here means around 1, 2, or 3% prevalence. If you look at those species don't make too much, sense in text, because these are species level genome beans that have not been characterized by microbiologists. So this is a very large set of microbes that are depleted in our population. We are studying why, but that is extremely strong. And the fact that they were there in the ancient population may mean that we lost these species after the Industrial Revolution, maybe. On the other side here, you see the species, and these indeed are quite well known because microbiologists were able to target them, that are highly prevalent in our McDonald's-style diet populations and are missing in the others. You may say you should confirm some of them, and indeed, this is one of the species we confirm. We isolated them. We are in the process of naming them, and this is important because we can study their functions. Of course, you may say, where did you isolate them? From ancient populations is impossible. For non-westernized population is possible. But these species happens to be also in API of my lab, and so we were happy to retrieve it. But it was present also in this very ancient guy that was found 5.3 thousand, that is from 5.3 thousand years ago, and was found 100 kilometers far from our lab in Trento. So this is the importance of looking at the unknowns. But coming to our population and coming back to the colorectal cancer study, we enlarged it quite a bit with several projects. Instead of having 7 studies, now we have more than actually we have 16 different studies. And when we redid the analysis at the level of species level genome beans, we found many different species of Fusobacterium nucleatum that are actually associated with colorectal cancer, and some are actually unknown species. So it is important to see these species also in our population. For example, when we look at the different stages of colorectal cancer, many different unknown species level genome beans are popping out, and these may be relevant in the progression of the disease. At the same time, I show you that it's important to look at the strain diversity. So this is important because many of us have different, for example, Escherichia coli strains in our gut, and this is the plot you can get if you look at the genetic diversity, genetic distance of 2 strains of the same species in different individuals. My E. coli, Daniel's E. coli is not going to be 0% genetic distance, they are going to be something like that, meaning that it's very, very rare to find two individuals with very similar strains. This can be repeated multiple times, and you may say, well, this is a very nice distribution of the noise. However, if you take my samples now and my sample 1 year ago, you will find the same strains. So, this means that our human microbiome is really unique at the strain level, even looking at one single strain, but it's stable in time. We keep our adapted strains in time, and this is, I think, very important. And this is again, another motivation for which we should look at the species diversity. And you can do that by cultivation. Of course, this is problematic, but what we do is doing through computational analysis. This is one of the tools that we developed for doing that, is looking at the genetic diversity of the unique marker genes I showed you before. So looking at single nucleotide variants in these species-specific genes that we retrieved. And if you do that, you see things like that. This is a Bacterium rectale. There were only 3 reference genomes from isolated sequencing a few years ago. And with these techniques, you can represent the diversity of the species. There are many different subspecies here. Some of them are really geographically adapted and different in different population and continents. They are also functionally different because this subspecies here is completely non-motile. There are 4 operons giving motility to this bacterium, and this subspecies here is completely lacking all of them. And at the same time, actually it acquired many other different genes for being able to do more general carbohydrate metabolism through single genes, but also through the acquisition of operons, very likely via horizontal gene transfer. Other species are similarly different. This is Protella copri, another bacterium for which we found that there are many different species of this bacterium. And this is one of the species that are highly prevalent in non-westernized population and are not prevalent in our population. And it's actually important to see that in non-westernized population, 2/3 of the individuals have all 4 types of Protella copri simultaneously present against all 4, or against 4% in our population. And this is probably due to the fact that this bacterium is degrading complex fibers that we are maybe losing in diversity of diet in our population. And again, this ancient friend of ours has all 4 types of Protella copri, meaning that maybe this is loss of diversity. And we can also put these strains, even ancient strains, in phylogenies, and this means that by strain level profiling, we can really uncover the phylogenetic diversity of the microbiome. All right, so I think I explained you what are our take-home messages on these 2 aspects, on uncovering species level diversity and strain diversity. But there is more, because if you, I told you that, well, I didn't tell you, but there was a slide on showing that on average, 2 different individuals, for example, in this room, may share around 35% of the species. I did show you a little bit more into the details that in terms of strain that are sharing is probably less than 1% for sure. And so the question is, how did we acquire this huge diversity in each of us? And we studied several years ago the fact that for sure the mother is transmitting strains to the infant at birth during the process of delivery and during the first few hours of life. But our adult microbiome is very different from the infant microbiome. So this may explain no more than 10% of the adult microbiome composition. There is food, fermented food is full of living bacteria. However, if you look at which of them are actually able to colonize our gut, is very little. I'm not saying that the diet is not influencing our microbiome. I'm saying that there are very few microbes from our diet that are actually colonizing our gut. And the same is if you look at other, environment, pets, your home environment, so on and so forth, less than 1% for sure. So, we need to hypothesize that there is a very, very deep transmission of strains here. And if you are familiar with the pathogens, and unfortunately we are all very familiar with some of the pathogens, there is a lot of transmission, and we need to characterize them also in the human microbiome. This is what we did with the strain level analysis, because if we saw that in two different individuals you have different strains. If you find two individuals with the same strains, well, that may be the result of transmission. And in this study, that is what we did, and we looked at almost 10,000 metagenomes. But the difference here is that we know the context of these metagenomes. We know all these metagenomes are from individuals that are interacting in some ways, maybe because they are living in the same house, maybe because they are twins, and so they were living together up to a certain point. They were mother infants, or they had other social interactions. When we did that, you find that indeed, again, it's confirmed, this is Bacteroides caccae, for example. You see strains from individuals that are longitudinally sampled, and the others have a very different distribution, the same thing I showed you before. And so this really allows you to put a threshold here. Whatever is on the left is same strain, whatever is on the right is a different strain. And we could talk a lot about this definition, but operationally, it works pretty well. So with this, you can look at transmission. So a meta-analysis here in infant, 1 day of life of the infant. Whatever you find in the gut from stool is actually 50% on average is coming from the mother. Is going down a little bit with time, with infants here that are 85 years old. So the mothers are centenarians, and you still see these transmission events. But this is probably more important. So this is the strain retention. Same individual, multiple time points, 90% of the strain is exactly the same. On the other side here, individuals in same or different population, not sharing in median, not even one strain. And actually 97% of the individuals in different population, they don't share even one strain. Here is mother-infant, around 30%, and this is coming basically from an average of the ages here. But here instead is the 15% of strain sharing of individuals living in the same house since a few years. So here, living in the same house means that you are sharing strains and there is a deep transmission network. We saw it in the gut, but also in the oral cavity. These are partners living together, and this is the oral microbiome with more than 40% strain sharing, compared against to 0 strain sharing in individuals that are unrelated one to the other. So this can lead to important conclusion. This is a paper not by us, but it was bringing this idea that some non-communicable diseases, if they are linked with the microbiome, and because the microbiome we show is transmissible, well, maybe it is at least partial. It is at least true that these non-communicable diseases are at least partially communicable. This is, of course, a provocative question, but there are at least some aspects that should be better investigated. I'm running a bit out of time here, but the same state transmission can be investigated when you look at fecal microbiota transplantation that is under study for many different diseases, as those that are shown here. And it's very important to understand whether so fecal microbiota transplantation, is just the transplantation of fecal material from a donor to a diseased individual, and it can reconstruct the gut microbiome of the recipient, although the recipient will have a microbiome which is a mix of the donor microbiome and its own microbiome before. And it's important to track the transmission of the strains to understand how much the microbiome of the donor engrafted in the recipient microbiome. You can see it, you can model it pretty well. These A and B are two different donors. The recipients are here in this gold color, and after the transplantation, if you look at the strain transmission, you can really cluster very tightly together the donors with the corresponding recipients. But it's not always so successful. So we have a strain engraftment rate which is very different in different conditions, including whether someone took antibiotics before the FMT or not. Important though, it is that strain transmission is linked with clinical success, so it's very important to characterize this. There are some species that are way more transmissible than others. I'm not going to go into the details here, but even more importantly, given the donor microbiome, the recipient microbiome, and the strain level patterns, we are able somehow to predict what is the composition of the microbiome after FMT. This is very important because you may use this approach to identify which is the best donor for a given recipient. I'm concluding here saying that if you are able to put together all these three levels of studies, well, you can have important clinical consequences. So these are some of the studies that we've been doing in just applying, but it was quite a bit of work, applying these techniques on specific problems: colorectal cancer, response to immunotherapy, engraftment of fecal microbiome transplantation, and personalized nutrition, also stratified based on the microbiome. I'm running out of time, so I am finishing here. Again, these are the three most important things that I think we need to uncover out of the microbiome to really move to relevant biomedical applications. Last 30 seconds, sorry, just to advertise that we have this new initiative. It's called the Microbiome Virtual International Forum. It's a completely free recurrent conference, 3 hours every month, both in the Atlantic and the Pacific time zones, in which we have abstract, keynotes, and highlights. And this is a completely open, free. You are welcome to join. We have a steering committee that can be also increased with any volunteers. And this is really something from the community, for the community, and is taking traction. This the second year. These were the fantastic keynote speakers of this year, and this is really growing. So I'm very happy if you want to join and ask me any question about that. We are very happy to involve you. Okay, with this, I'm stopping here. Let me just take a few seconds to thank all the members of my lab because they did all the work that I show you today. And we also have openings, so get in touch if you are interested in this kind of work. And thank you so much for your attention.
So we have time for a couple of questions. I'm just going to start here.
Very interesting talk. Thank you. So when looking at the microbiome transmission between people in the same household, how do you distinguish between actual transmission and maybe they're just eating the same diet and so there's some determinism there?
Yeah, so that's a great question. So diet has a lot of impact and really for sure there is a diet effect inside the same house. But the diet acts on the level of which species are present and what the abundance of that species. Diet cannot give you directly new strains because it's very rare that the conditioning food are actually the conditioning gut. So gut microbes are 99.9% present only in other guts. So it's a combination of transmitting strains, and then maybe diet will dictate which of these strains are a good fit for you and which are not. But without someone donating somehow you the strains, you will not be able to enlarge the gut microbiome because really is only in other guts you find the microbes for your gut.
Thank you for the great talk. I totally agree that string level analysis is very important, but I always have a question about the resolution when you do the string level analysis, because there's so many strains such as E. coli, and some of them are very similar. So some tools, when they do the string level analysis, they will do a clustering using a sequence similarity cutoff. But some strains, even if they are very, very similar, they might have different biological properties. So I want to hear your perspective about what's a cutoff or resolution we should do the string level analysis. Thank you.
Yeah, so this is a great question. So many tools are doing subspecies level analysis. I would say is not strain level analysis. And so the question can be probably reformulated based on what is a strain. No, so in some cases, two genomes with only one SNP may have different functional characteristics because of that single SNP. In other cases, maybe there is quite a bit of variation, but the strains are almost exactly the same. So what we did in our transmission analysis was to adopt an operational definition of strains. For us, a strain is the same strain if it is very similar inside you in 5 to 10 months distance. So basically, the distribution of distance, if unless you had a strain replacement, there are very few SNPs in, again, a time frame of 5 to 10 months. Maybe I said yes. Sorry, I want to say months. Instead, if you look at different individuals, you see a very different distribution. For us, what is the same strain is the threshold put between these usually separated distribution, which usually means from 50 to 200 SNPs at maximum.
Okay.